Hi, I am Muhammad Yusuf bin Ismail.
I am Muhammad Azhar bin Muhammad Syamsuddin.
My name Amir Ashraf bin Muhammad Azman.
Immunological techniques include both experimental methods to study the immune system and methods to generate or use immunological reagents as experimental tools. The most common immunological methods relate to the production and use of antibodies to detect specific proteins in biological samples. Via cellular, molecular and genetic modification, this method enables immunologists to modify the immune system. These methods are not limited to the field of immunology, but are commonly used in many other biological disciplines by basic scientists and in human veterinary medicine by clinicians. These are the top 7 types of immunological technique used in biochemistry. First, immunoassay, radioimmunoassay, enzyme-linked immunoadsorbent assay, immunoprecipitation, immunoelectrophoresis, immunofluorescence, and the last one is immunohistochemistry. The most immunological technique available are focused on the study of the adaptive immune system. In this explanation, we will be focused on the two immunological techniques, which are immunoassay and immunoprecipitation. An immunoassay is a biochemical test that uses an antibody and usually or an antigen to determine the presence of concentration of a macromolecule or small molecule in a solution. Burson and Yellow in 1959 identified the first immunoassay. Their research resulted in their receipt in 1977 of the Nobel Prize in Medicine. Immunoprecipitation is the process of using an antibody that precisely binds to the particular protein to precipitate a protein antigen out of the solution. To isolate and concentrate a single protein from a sample containing several thousands of different proteins, this method can be used.
So now we are going to talk about the type of immunological techniques. The first one is immunoassay. Immunoassay can simply be defined as a procedure of measuring or detecting a specific protein or substances using their properties as antibody or antigen. It can also be defined as a biological test that uses to measure the concentration of substances in biological liquid, typically serum or urine, through the reaction of antibody to its antigen. The assay takes advantage of the specific binding of an antibody to its antigen. Immunoassay uses antibodies, which is an immune system that naturally create to detect germ. It uses specially designed antibody, which uses to find specific chemical, and it will be held into for the rest of the time. So immunoassay has a specific structure, which is two binding site for antigen and one binding site for the substances that we are going to find and investigate. So immunoassay works by three complex, which is the first one is the antibody itself, the second one is the analyte, which is the substances that we are going to analyze, and the third one is the label, which is to detect the whole complex. The result of immunoassay will be shown depending on type of immunoassay being used, either through color, light, or other signal. The main purpose of immunoassay is usually to check on the contamination or a diseases, but it also can be used in a pregnancy test, drug test, or a biological research.
Immunoprecipitation is the small-scale affinity purification of antigen using a specific antibody that is immobilized to a solid support such as magnetic particle or agarose resin. And then, this technique used in many fields of science that isolate functional protein using the high specificity of antibody. Okay, we go to principle. A protein complex can be isolated from a protein mixture by using an antibody that is specific for one protein of the complex. For your information, there are three main steps involved in this principle. For the first, incubate sample with antibody against protein of interest, and then this antibody protein complex is spread from remainder of the sample isolate, and lastly analyze. You can see the form of immunoprecipitation reaction. This. Okay, so immunoprecipitation have two major methods, such as direct capture method and indirect capture method. Let me conclude the both of method. Basically, both method give the same end result with the proteins or proteins complex bound to the antibodies, which themselves are immobilized onto the beads. Okay, that's all from me.
In conclusion, immunoassays have been widely used in many important areas of pharmaceutical analysis, such as diagnosis of disease, therapeutic drug monitoring, clinical pharmacokinetic and bioequivalence studies in drug discovery and pharmaceutical industries. The important and widespread of immunoassays method in pharmaceutical analysis are attributed to their inherent specificity and high throughput and high sensitivity for the analysis of wide range of analytes in biological samples. These are the list of references that we use to find the information about the immunological techniques.